question number 11. Read the statement A and B. Select the correct matching option. Statement A. Bulliform cells are present in abaxial epidermis of grasses. Dear friends, you know Bulliform cells are also called motor cells. They are found in the upper epidermis or dorsal epidermis of isobilateral leaf. Grass is isobilateral leaf and upper or dorsal epidermis is called adjoining epidermis. But here it is mentioned abaxial, thereby statement A is incorrect. Now we come to statement B. They are large, empty, colourless cells which under water stress condition make the leaves to curl inward. I hope all of you know this is correct because motor cells are highly enlarged, thin-walled, empty cells. When they become turgid, leaf is going to open, and when they become flaccid, it causes rolling in or folding in of leaf. So A is incorrect, B is correct. Option 3 is the answer. Also, I want you to remember that Bulliform cells which are found in isobilateral leaf of monocot is an adaptation to check excess of transpiration under water stress conditions. Now we move on to the question number 12. Cell junctions that facilitate the epithelial cells to communicate with each other by connecting cytoplasm of adjoining cells for rapid transfer of ions, small molecules, sometimes big molecules are. Let me go through the options. Plasmodesmata, I hope all of you know, plasmodesmata is found in plant cells, thereby it is not an answer. Tight junctions, I hope all of you know, tight junctions are the one which are non-leaky junctions, thereby they do not allow anything to move. So even option 2 is not an answer. Option 3, adhering junctions. I hope all of you know adhering junctions are cementing junctions that help in attachment. Thereby even that cannot be an answer. Option 4, gap junction. Yes, gap junction is the answer because it is this which is going to allow the molecules to pass through it. Now we come to question number 13. Fertilization in earthworms occur in. I hope all of you know earthworm is an annelid, an oligochaete where setae or chaetae are locomotory structures, and here it is related to fertilization. First you should know it is bisexual animal where cross fertilization is a role. I hope all of you know two earthworms come together and exchange the male gametes in the form of spermatophores. Please remember, bundle of sperm is called spermatophore, and there are 4 pairs of spermatheca present, that is in segment 6, 7, 8 and 9. And also remember there is a 3 segment fused undifferentiated region called clitellum, 14th, 15th and 16th segment, and this region is rich in glandular cells that secrete cocoon into which the egg is put. As the egg is carried through spermatheca region, spermatophores are released and fertilization occurs in the cocoon, and then the cocoon is going to be dropped into the soil. I hope all of you know normally it is going to carry 2 to 20 embryos that develop into earthworm. So 2 to 20 babies. Now we look into the option spermatheca, no, moist soil, no, oviduct, no, cocoon, yes. Now we move on to the next question. Choose the correct statements. Ribosomes are found only in cytoplasm and also on rough endoplasmic reticulum. I hope all of you know ribosomes. Ribosomes are going to be produced in nucleosomes in case of eukaryotes. So they also occur in nucleus. Thereby this statement is incorrect. We have to pick the correct statement. So option 1 is not an answer. Centriole is a membrane bound organelle found in animal cells that help in cell division. Remember, centriole is non-membrane bound organelle. Thereby, even this is wrong. The genomic DNA of prokaryote is single linear chromosome. Genomic DNA, chromosomal DNA in case of prokaryote is single, correct, but it is not linear, it is circular. Thereby, option 3 is not an answer. Option 4, inclusion bodies are non-membrane bound reserve material aggregates found freely in the cytoplasm of prokaryotic cell. I hope all of you know this is correct answer. Please remember phosphate granules, synophysian granules, glycogen granules, all these are examples for the cell inclusions. Now we come to question number 15. The non-staining secondary constriction seen at a constant region in few chromosomes are called. So here I read the question again. The non-staining secondary constriction seen at a constant region in few chromosomes are called. So remember, secondary constriction regions they are asking, and after that we call it as satellite, and such a chromosome is called as SAD chromosome. SAD refers to sine acidothymidine, which is RNA rich region. Here thymidyllic acid, a constituent of DNA, a nucleotide of DNA is absent. Let me look into options now. Option 1 says kinetochores. Remember, kinetochore is plate-like structure that are found in primary constriction region. Option 2 satellite, that is our answer. Option 3 nucleosomes. I hope all of you know they are ultrastructural unit of eukaryotic chromosome. Each nucleosome is made up of around 200 nucleotide base pairs wrapped over histone octamer. I hope all of you know histone octamer is made up of a pair of H to A, H to B, H3 and H4 histones. Last one says heterochromatin. I hope all of you know when we stain prophase chromosome, we are going to see certain regions deeply stained, certain lightly stained. Deeply stained are genetically inactive regions and are called heterochromatin. Light stained regions are genetically active regions because they are less condensed and they are called euchromatin. The differential staining property of the chromosome due to the presence of heterochromatin and euchromatin areas of a prophase chromosome is called heteropyknosis. Now we move on to the next question. Question 16. Study the four statements A to D given below and select two correct ones out of them. A. Apoenzyme are protein portions of complex enzymes. I hope all of you know when you talk about enzymes, complex enzymes are made up of protein part and non-protein part. The protein part are called apoenzymes. Thereby, statement A is correct. Second, prosthetic groups, coenzymes and metal ions are the cofactors. Yes, protein part, apoenzyme, non-protein part is cofactor. If cofactor is organic, then it can be a coenzyme or prosthetic group. If the organic constituent of a complex enzyme, which is a cofactor, is going to associate during enzyme action, we call that as coenzyme. And if it is always associated, means it is a part of it, it is firmly fixed to it, that is apoenzyme, then it is called as the prosthetic group. So, statement A, statement B are correct. We have to pick the two correct options. Thereby the answer is option 3, which says A and B. Then we come to next question. The polymers of fructose is. Please remember fructose polymer. Let me look into this. First option says cellulose. Cellulose is a homopolymer of glucose. Thereby it is not an answer. Second, chitin So remember, chitin is a homopolymer made up of N-acetyl glucosamine. Thereby, even it is not an answer. Remember, chitin is cell wall material of fungi and body wall material of arthropods. Third is glycogen. Glycogen is a homopolymer of again glucose. The difference between glycogen and cellulose is glycogen is going to have secondary chain and thereby iodine can bind to it, thereby glycogen is going to test positive to iodine test, whereas cellulose do not show positive result to iodine. So also when you talk about starch, starch also show positive test to iodine. Last one is inulin. Please remember it is inulin, not insulin, which is a polymer of fructose, and thereby option 4 is correct. Now we move on to question number 18. The ash left after complete burning of the tissue does not show. So when we are going to burn the tissue, what is going to be burnt? So remember, for burning oxygen is required. Carbon dioxide, carbon combines or burn with oxygen to become carbon dioxide. Therefore, carbon will not be present in the ash. With this, we look into the options. Option 1, potassium. Option 2, calcium. Option 3, carbon. Option 4, magnesium. So we know carbon as the answer, thereby option 3. Now we move on to the question number 19. During the G1 phase of cell cycle, if the DNA is denoted by 2C and number of chromosome 4, by the end of S phase, the DNA and number of chromosome would be. Please remember, G1 phase is the post-mitotic pre-replication phase, and during S phase replication occurs, and as a result of replication the DNA amount is going to increase, and we are going to have each DNA represented by sister chromatids, whereas the sister chromatids are held by the same centromere. Only during mitosis splitting of centromere occurs at the beginning of anaphase. Thereby there will be doubling of DNA but not doubling of chromosomes. Chromosome number remains the same. So what is the answer then? When you look into the option, option 1 says 4C and 4. Thereby option 1 is the answer. Now we come to question number 20. Again, match the following type. Column 1, here we find certain statement related to the events that happen during first phase of meiosis. Column 2 has given 5 substages seen during first phase of meiosis 1. And let me look into it. Separation of bivalents. So I hope all of you know, separation of bivalents happen during diplotin stage. Thereby A is matching with 5. Formation of tetrads. So remember, tetrads refers to bivalents, and this is going to happen during zygote event, because point to point pairing of chromosome occur, what we call as synapsis. So formation of tetrad match with zygote, that is 2. Crossing over. Crossing over relates to crossing over of non-sister chromatids between homologous chromosomes, and it happens during packaging. So C is matching with 4. Finally, chromosome compaction. I hope all of you know, the first stage of phase 1 of meiosis 1 is called as leptotin stage, which is bouquet stage, where orientation of the chromosome occurs, which is called as condensation, and orientation, which is called as chromosome compaction. Thereby the answer is option